Okay, so we have a lot of cells, and these cells need to communicate with one another. And the way they do that is through a bunch of different receptors and signal molecules. So the less common kind of receptor would be the intracellular receptor. So for that kind of receptor, you would have signal molecules that are small and hydrophobic so that they can get through the phospholipid bilayer easily and then bind to these receptors inside the cell and set off another signaling pathway. You can also have cell surface receptors. There's three main types that we'll focus on. So first, there's the G protein coupled receptors, or GPCRs. So that would be this blue thing over here, and then they are coupled to this G protein, which is a trimeric protein made of the alpha unit and the beta and gamma subunits. So this signaling molecule is going to be larger and more hydrophilic, so it will stay on the outside, doesn't actually ever enter the cell, but it activates this GPCR, which then activates the G protein, separating the the beta gamma subunits from the alpha subunits. There's also the ion channel coupled receptors, or ligand gated. So thanks to these ligands or signal molecules binding to them, it opens the channels, allowing for the ions to get through. And lastly, we have the enzyme coupled receptors, which are either enzymes themselves or are associated with other enzymes. So the signal molecule, usually in the form of a dimer, comes along, and then these two will join together and then activate either themselves, the enzyme part of them, or the other enzyme associated to them, continuing the signaling pathway. And all these things will be used to change the cell's behavior. So that could be, say, working with the proteins that you already have, so altering their function to make them work more or work less. And you can also change gene expression itself to make more of whatever protein you want to make.